Welcome back. I'm on the wrong slide. I'm way back, aren't I? Ooh, okay, there we go. Welcome back. We're going to be talking about the nucleus and its function. So the nucleus is the powerhouse of the cell. It is the brain of the cell. It's basically the region of the cell that contains your genetic information, and that genetic information is then going to be used to produce new cells. So it carries all the information that makes you who you are. Within the nucleus, we have a lot of DNA. So all these little tiny squiggly things, those are DNA. When it's uncondensed, it's called chromatin. When it is condensed, it's called chromosome. And then in the very middle, we have the nucleolus, which is where ribosomes form. In the human, most cells are singly nucleated, so they have one single nucleus per cell. But there are some examples, and the best example is the skeletal muscle is multinucleate. So here we have one single muscle fiber highlighted, and you can see there's one nucleus, there's another nucleus, there's a nucleus, there's a nucleus, there's a nucleus. So there's lots of different nuclei within this one single cell. You can see them up here as well. So skeletal muscle is multinucleate, whereas blood is anucleate, so it has no nucleus. Blood cells are packed so fully with hemoglobin that prior to maturation, they expel the nucleus and degrade the majority of their organelles. So they basically are just a spherical structure that contains hemoglobin. And they move around our system carrying hemoglobin, which binds oxygen, and they bring the oxygen to our cells all the time. That's what they do. So skeletal muscle is multinucleate, whereas blood is anucleate, or has no nucleus. So inside of the nucleus, we have this nucleolus. The nucleolus is a region of the nucleus that produces ribosomes. Ribosomes are composed of protein and RNA. I haven't talked a lot about the ribosomes yet. Ribosomes function in protein synthesis. They are formed from a small and a large subunit, and they are basically the workhorse of protein synthesis. So they make proteins. Some cells have more than one, or some nuclei have more than one nucleolus, whereas others have no nucleolus whatsoever. So within the nucleolus, or within the nucleus, not nucleolus, I'm sorry, we have DNA. DNA is found in different forms. So when it is uncondensed, it's called chromatin. And when it is all condensed up, it's called a chromosome. So I like this picture, this figure I found off of Google Images, because it's like spaghetti and meatballs. You take the spaghetti and you wind it around the meatballs or the histone proteins, and you form these condensed chromosomes, which is what most people think of when they think of DNA. So what does DNA actually look like? Here you have, again, the helical structure. It's wound up around these histone proteins. If we move it further and further out and we unwind it, you can see that DNA is a double-stranded molecule. It contains the backbone, which has the sugars. In DNA, it's deoxyribose and the phosphate. These are formed, or they form or bond together in what we call phosphodiester bonds. And then they have the nitrogenous bases, adenine, thymine, guanine, and cytosine. And adenine and thymine always bind together. Guanine and cytosine always bind together. And they bind together through hydrogen bonds. So the two single strands here and here are bonded together, forming that double-stranded molecule via hydrogen bonds. DNA condenses up anytime we're going to get ready to divide. So it's important that you keep all of the same strands of DNA together. So you have 23 DNA strands from mom, 23 from dad. Each parent gave one of each DNA molecule, or one of each chromosome. So we have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s. And so those chromosomes, or the DNA, condenses, forming the chromosomes, and then those chromosomes come together so that we don't end up with too many chromosomes in any given cell. So how does DNA actually replicate? There are 3 basic steps. We have unwinding of the DNA. Then we have to break the parent strands. So you're breaking those hydrogen bonds. And then we're going to allow new DNA nucleotides to come in and build a complement to each of the parent strands. That's the basic process. To do this, we're going to need DNA polymerase, and we're going to need some type of molecule like topoisomerase to unwind the DNA. DNA polymerase, once the DNA is unwound, will bind and produce complementary strands to the parents. So if this is the parent strand, the complement would be TTAGC, TAGC, because it's complement to this strand. Then we bind these together, make hydrogen bonds, and DNA ligase comes in and seals everything together, so on the backbone, and you end up having a complement strand here, daughter strand to the parent strand, and that is termed semi-conservative replication. So here is actual replication. You can see it. Here we have DNA polymerase. DNA polymerase binds to both sides. One of the strands is going to be called the leading strand. The other is going to be the lagging strand. The leading strand allows nucleotides to come in and just continuously bind and form new bonds. The lagging strand does not allow that. And so the difference is on the leading strand, DNA can only be produced in a 5' to 3' direction. And if you look at DNA, right here you can see we have a 5' to 3' direction here, but down here they're going to be opposite. So if this is 5, this is 3. And then down here you have the 5 and 3. If you unwind them, one side, so here's the 5, it unwinds down to here to this side to the 3. This is the 5 over here. It goes up to the 3. So they're anti-parallel. You can only add DNA to the 3' end. So you're adding to both sides, but when you unwind it, one side is going to be the 3' end, the other side is going to be the 5' end. And if it's the 5' end that you have opened, you're going to bind over here and build in the opposite direction. And then you're going to have to bind here and build in the opposite direction, and then bind here and build in the opposite direction. Whereas the leading strand is just continuously being produced. And I do have a video. Oops, sorry. While this is doing its thing, I'm going to get rid of some of those.
If you write anything on your computer, you need to get Grammarly.
Oh, that's not what I wanted to do. Sorry.
I write pretty much all day, every day, and Grammarly makes my writing better.
As a student,
There we go.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
Hold on. I shouldn't have done that because it's going to start a new video shortly. I already know that. There we go. It always does that to me, and I'm not going to, that just drives me crazy when that happens. So now I have to find the place that I was at. Oh, that's, okay, here I'm at. All right, so if you have any questions about any of that, definitely bring them to my class. So not only do we have to replicate the DNA so that cells can divide, but DNA also codes for all the proteins that make you who you are. And so a protein is also known as a gene. And that's just a region of DNA that codes for specific proteins, right? A gene is a region that codes for specific proteins. Genes are going to have promoter regions, and they're going to have terminator regions. A promoter region is going to be where the gene starts transcription, and then the terminator region is where we stop it, okay? So gene expression or protein synthesis has two basic processes, transcription and then translation. Transcription takes DNA, which our cells can't understand. It's like having a boss that you just don't understand. And so that boss has to tell someone else, hey, this is what's going on, and then that person can bring it to you. So maybe it's like the boss to the foreman to the workers. That would be an example. Transcription is the formation of messenger RNA, or no, I won't say that, is the process of forming an RNA transcript of the DNA. RNA can be read and understood by our cells, but DNA cannot. Transcription occurs in the nucleus. Translation then is the process of taking the RNA transcript and producing the actual protein. That occurs in the cytoplasm on ribosomes. There are 3 types of RNA that can be formed during transcription. The one we always talk about is messenger RNA, but we also produce our transfer RNAs and our ribosomal RNAs through transcription. Transcription has 3 events, just like your replication. You have initiation, elongation, and termination. So here you see the DNA molecule open up at a region where there's some type of protein being coded for. The RNA then, RNA polymerase, can bind, and RNA polymerase allows the RNA nucleotides to come in and complementary bind to that DNA molecule. Once the RNA polymerase has coded or has produced the entire transcript, then the transcript is going to move away and the DNA molecule can be rewound up. This is my example, and I know in humans we are eukaryotic organisms. This is an example I took from my microbiology lecture that I do, because transcription is very similar. Transcription and translation occur, I won't say that. Transcription occurs in a 3' to 5' direction. So you are always adding to that 3' end and producing in a 5 to 3' direction. And so either strand of DNA can be the strand that codes for a protein. In this case, this is our coding strand, and this is our template strand. We use the template strand, which is the opposite the coding, to produce our transcript, because the transcript then will be identical to the coding strand, except for wherever you see T's in the actual coding strand, you'll have a U, because remember, RNA, it's single-stranded, and it does not contain thymine. It only contains, it contains adenine, guanine, cytosine, and then it contains a uracil. Let me just add those. Okay. So here we have the promoter region would be right over here, and then you have this little region here that's being produced, but it's not actually coding for anything, and then you have the actual functional amino acids or messenger RNA being produced. But in eukaryotic organisms, so humans, our messenger RNA has to be modified. Our DNA doesn't just code for proteins. And I always like to ask how much of the DNA actually codes for proteins. And it's amazing how many people, or what people think, because a lot of times people just assume that 100% of our DNA codes for proteins. What else does DNA do, right? But it's not true. I'm not going to tell you how much of our DNA codes for proteins, but I'll probably ask it in class and see how many of you can figure it out before class starts. So because not all of our DNA codes for proteins, when you produce that messenger RNA molecule, it's not actually considered a mature molecule. It has to be modified before it can leave the nucleus and enter the cytoplasm. So the first thing we have to do is we have to cut out regions that don't code for the protein. So regions called exons are the regions that code for protein. They express the protein. Introns are regions that don't code for the protein. We have to cut out all of these regions called introns and splice together these regions called exons. And then we have this protein coding messenger RNA. So we use spliceosomes to do so. And so this is what a spliceosome looks like. It basically takes one exon and another exon, and it takes out the intron and splices those two together. Once we do that, we then can add a guanine cap. Guanine cap is just what it sounds like. It's a bunch of guanines all lined up that we put at the beginning of the messenger RNA, and then we add a polyadenine tail or a poly-A tail at the end. Once we've done that, we now have a mature messenger RNA. So the cap and the poly-A tail have important functions as well. The transcript that we've just produced has to leave the nucleus and enter the cytoplasm. In the cytoplasm, we have a lot of dangerous proteins that will break down any type of nucleic acid. So the guanine cap and the poly-A tail protect the messenger RNA from these proteins, but the poly-A tail also functions to keep the ribosome from falling off too soon and not producing the entire protein. Now that we have the messenger RNA, we can leave the nucleus and through those nuclear pores and enter the cytoplasm or the rough ER and attach to a ribosome. So I'm going to stop here. I know we just started protein synthesis, but I've gotten through the first half of protein synthesis, and this is a 21-minute lecture, and I want to kind of keep my lectures relatively short. If I make them too long, people get tired and they don't want to listen to the whole thing. So I know this. So I will talk to you in a little bit. Bye.